recording. So we have, you had an activity. If it is turned in already, you got points. If it's not turned in, after you turn in after this period, if you turn in after I explain the answers right now, it's minus 10%, minus 20%. If it's not complete, you're still losing points, but it's better than minus 20. So if you want some decent credit, I need to see decent amount of work done. So I'm just letting you know, I'm going to give you 5 minutes to get that done. I recorded this to make sure everyone understands you had a fair opportunity to turn in. You should have been done already. You should have scanned it. You had till yesterday at 11:29 p.m. to scan it and upload it. And I'm giving you the opportunity to do it now for another 2 minutes, and then we're done. You're welcome, by the way. These ideas are are deadlines to you suggestions? When somebody says to you, this is due today, that means, what does that mean to you? Maybe it'll be due. Maybe I'll turn it in later. What does that mean to this class? You have to turn things in. There's a deadline. You're losing 20%. Every day it's late. Turn in tomorrow, it's minus 20. I'm giving you an opportunity to turn in today for full credit. Get it done. You got another minute. The fast 7. We don't have time for me to give you extension after extension. So, all right. So at this point, we're going to look at this. This is called simple diffusion. We talked about it. We drew it in class on Monday. You have these molecules. They're bouncing around, and they say, how many different types of molecules are shown in Model 1? I think it's obvious that the answer is 2. All right. Count and record the number of triangles and circles found on each side of the membrane. On the right and versus the left, you have circles versus triangles, circles versus triangles. How many circles on the right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Do you agree? 12. How many circles on the left? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. And 13 on the left. Is that correct? Triangles, 0 on the right and on the left, 1, 2, 14. Thank you. I'm going to go ahead and assume you're right and move on. So that's how many. Just count. I don't think that's difficult. So let me write this clearly for a second. We're talking about a system, if we look at this simple diffusion, that has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Do you agree there's 13 here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So there's 13. 13. Out of what? Out of 13 are circles. 13 out of 13 are circles. On this side, you got 1, 2, 3, 4, 5, 6, 7, 8, 9, 3, 4, 5, I'm going to count again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Here, this side is 11 out of what? 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25. 11 out of 25. Right? There's 25 molecules on this side. 11 of them are the small circles, right? Do you agree? So the concentration on the left, this is the left, versus the right, the concentration on the left is 11 out of 25. So this on the right is high, and this on the left is low. Even though there's less circles, correct? It's a less number. Now, there's it's less concentration, even though there's and there's also less circles. So either way, it's low and high. So this is high and this is low. Why? Because concentration is equal to the amount of solute divided by the amount of solvent. Again, review, you should know it, covered it on Monday and before. We had mentioned this earlier in the year as well. So on this side you have high and this side low. Now the triangles, the first question I told you on Monday you had to ask was, the first question number 1 is, does the solute cross, not even solute, I shouldn't even say solute, does the molecule cross the membrane? That is the question. There's the question. Does it cross the membrane? Triangle? No. Circle? Yes. So now, in what direction would you think that this circle's going to move? Remembering, this is right and this is left. What direction is the circle going to move? You say it's going to move to the right? So that you're saying this circle here, just to make sure I understand, this circle is going to move in this direction. You're really good point. I love your thinking. Yes, you're right. And go either way. And go anywhere. They'll go that way and go this way. But net. Let's go ahead and review that concept as well. Net. When you talk about net movement, net. What does net mean? That means net movement in this case is equal to the movement in one direction minus the movement in the other direction. Now, what does this word net mean? Let's be clear on what this word means. The easiest example I have for you is an example where a person, some student, one of you, goes to work and makes $1,000 per week. Now that's how much they're gaining. So let me put it this way. This is what they're gaining in that week because they're working hard. Is there a reason you're on that laptop? On these, why these laptops and iPads are open? Are you writing? You're writing your notes. And you back there, is there a reason your laptop's open? Yeah. Okay, could you close it? I'm asking if you had a reason. Just I want to make sure you're paying attention. Why are you looking with that frown on your face? Yes. Okay, just asking. Look like you have, give me a frown. I don't know. So this is your work, so much you're gaining. But you realize that you don't just gain when you go to work, right? Because you're also losing money when you buy Starbucks and you eat lunch. So that's right. So about $50 a week for food at work. And then you have to minus probably what? If you live close, $10 a week for gas if you live close. Just to go to work, of course, so you can probably spend more than that. Then you probably, don't forget taxes. Why don't we put the minuses in red? That's what they like to do in accounting, right? They put minuses in red. So $50 a week, $10 a week for gasoline. And then minus taxes, and I have $1,000. You guys in a lower income tax bracket. Let's say you're $80. You're losing more than that. You're gonna lose more than that to state, federal, whatever tax. I'm just making these numbers up. So that your net, we call this the net. Wow, very nice. God help you in the real world, man. They're gonna get so upset. It's okay. You don't have to apologize to me. It's something that's gonna hurt you, so you should think about it. But right now, we're at 90 plus 50, which is what? $90 plus 50, 140, which leaves you how much money? How much money did you actually make? 1,000 minus 140. $860 is your net profit per week. So you're going to go get a job, and they're going to tell you $1,000 a week, but really you're only bringing up $860. And so when we talk about net movement, it's the same idea. You do have molecules moving in both directions. You're right. They're moving in both directions. But when you have a higher constant, when there's mostly on this side, and they randomly move in this direction, mostly on this side, they're a smaller concentration movement. Which side do you think is going to move? Are you going to move more towards? Well, the rule is, and it's one of those things I told you got to know, the rule is you move from, it's a rule that covers everything, including the wind, electricity, and diffusion and osmosis. Things move from high to low. High to low. All things went from high to low without extra energy. Now, people were like, well, some people were like, this is hard. Well, you're right, it's hard if you did not listen on Monday and make sure you knew the things I told you were going to need for Wednesday. I told you were going to need these things on Wednesday. I posted the video among other videos. I said, you need to know this before you even walk in here on Wednesday. You had Tuesday, the rest of Monday, you had Tuesday. We covered it in class. Wednesday, you showed up and you tried to do this without remembering that things move from high to low, without remembering what the word net means, without remembering that you have to ask this question first, and without remembering what is concentration. So, of course, you couldn't do it in the time you had. You didn't do the prep work. I keep telling you, you got to do your stuff before you walk in. Yes.
But the high to low is concentration?
Yes. High to low concentration is correct. So the net movement initially is moving happening in both directions. Yes. But mostly it's going to be moving to the left.
I have a question.
Yes, go ahead.
For number 21 is the answer in that little reader box.
Can we answer 3, 4, 5, 6 first, real quick? So 3 is which shape is larger, the triangle, correct? Now I'm just going to answer. I explained it. Now I'm just answering. Describe the direction of movement of molecules in both directions. The circle is moving in both directions, but mostly, or net movement to the left, where it's less concentrated. There's talking going on, people not focused. I'm okay with that. Please don't complain to me that this is hard then. Which molecules are able to pass through the semi-permeable membrane? Small circles. That's all you know. Somebody close that door. So if you left this system on it for a long time, what would you expect to happen? I hope you understand that what one would expect. The right side, which has a higher concentration, would have the small circles, small round molecules move to the area with less concentration. The left. So we'd have a net movement from right to left. Net. Of course, what we like to do is draw a big arrow moving into the left and a little arrow moving to the right to demonstrate that you have these small circles are mostly moving in larger numbers to the left. Now, what's a rather good example for this? Let me explain this and see if it helps. I give you these examples and then you try to answer questions. If you're not paying attention to these examples, if you're not understanding the examples, then when you go to try to apply them on a test, you're going to be confused because you don't know what's going on. I can't say it any clearer than that. So if you imagine there's about 30 of us in here. And what if you started walking around like zombies, just walking around and bumping into things, randomly moving in every direction. Every time you hit something, you bounce and move in the opposite direction. Can you imagine if there's that door were open, that eventually some of you would wander right out the door? Yeah. Right? You can imagine that, correct? Yeah. Now, what if the 30 of us were walking around here like zombies, bouncing in every direction, some of us were leaving, but outside you had 2,000 kids, people, also moving in every direction? Okay. How many people do you think would be right in the same space? All in this small hallway right here outside our door, you fit 1,000 people. What happens if they're moving around, bouncing around, and you open that door? Do you think more people would come in here or more of us would leave? More people keep coming in here until we're both about even numbers. Then people will be bouncing around randomly in both directions equally, correct? But that's what we're talking about here. The one that's higher concentration is this one. The one that's lower concentration is this one. So there's more of them here. So you'd expect that they would bounce in this direction more. Does that make sense? All right. Now let's take a look at this membrane here. Now you have a small nonpolar molecule, and then you have a polar molecules. They are these little dots. There they are. Which are the only ones crossing the membrane? Small polar, small nonpolar. If you look at the notes on Monday, I clearly told you, if you didn't get the notes, you were absent, you didn't write them down. Children, I posted the video. Go look at that section and copy those rules down. Small nonpolar molecules move through the membrane without any problem, period. You need to memorize that. You need to know it. So then, of course, you have your phospholipids here. They look like this, just like the ones you made in the model. Of course, you didn't do the model. Then you don't understand what's going on. Then you have some surface proteins here. And then you have transmembrane or membrane spanning proteins. So proteins that go across the membrane. You built them. You built them in this model. You should have if you did it. See, some, like these two, don't cross the membrane. They're surface proteins. But this one would cross the membrane. So some cross, some don't cross. Thank you for bringing your model today. So you see there's two kinds of transmembrane proteins here, or membrane spanning proteins. Then there's a carbohydrate chain. That's what this thing look here. That looks like a tree. That means a bunch of sugars covalently bonded to one another. Some are attached to proteins. Some are attached to lipids. Take the hoodie off again, Adam. The ones that are attached to proteins, we call them glycoprotein. The ones that are attached to lipids are called glycolipids. It's just that simple. All you had to do here was read. And there's a little bit of interpretation for understanding what the word glyco means. But you should know glyco because we talked about it before. Glyco means sugar. So it's sugar protein and sugar lipid. Okay. So that being said, let's answer the question. What two major types of molecules compose the majority of the cell membrane? I hope it's obvious to you that the majority of the cell membrane is made of lipids, specifically phospholipids, and proteins. That's it. That's all there is to it. And some people put transmembrane proteins or lipid spanning protein, membrane spanning proteins. That's fine too. But proteins is the point. How many different protein molecules are found in Model 2? Look, guys, it says protein, it says protein, it says protein. So these are you could say 4 plus 1, 5, right? Or you could say 3, 1, 2, 3, 1, 2, 3. Or you might have even said 2. It depended on how you looked at the problem. But I'm going to go ahead and say 3 because you have small surface proteins, membrane spanning proteins, and you have glycoproteins. But any of those other answers would be fine because at least you're reading the word protein and you're counting them. You might have counted this as 4, 1, 2, 3, 4, whatever. I'm good with you just looking and proving that you've read it and you can. It takes a little more interpretation to say 3, so it's a better answer. But I don't think any of that was too hard. What is the difference between the position of cell surface proteins and membrane spanning proteins? I think, again, it's pretty evident, right? If you have, and it's something that we've drawn on Monday. You see that I drew it all on Monday on purpose. You have a cell. Hold on a second. You have cell surface proteins sit on the outside, or they sit on the inside, and then you have the spanning proteins. They go all the way across, and they have two different purposes, right? One's to let things in and out, or to signal, whether it's a signal or molecules. Either way, it's gonna let things in or out. And the others on the cell surface, their function can only impact out here. And this one's function can only impact what's inside, right? It's like having a telephone outside the room without doors and windows. It does us no good because we can't get to it, right? Yeah.
Question about the last, with the model.
Yeah.
So
Wait, what's the last one? The last question?
No, it said, you know the model 2?
All right, hold on. All right, what's your question again?
Okay, so you see how some of the proteins are trying to shove its way into the phospholipid or the bilayer?
Here, you talking about this one?
No.
This one?
That one.
This one here?
Yeah.
I see.
Why is it trying to, why does it?
It's not really, it's just embedded in, you see how this is a hydrophilic protein. It's one that likes water. This one likes water. Where is it? Is it in the lipid layer? No, it's in the head. It's with the heads. It's hanging out with the heads. It's either covalently bonded or maybe it's just dissolved by the lipids. But either way, there's not really, just that they turn the membrane in just the way they drew it. So there's no real difference between this one and this one. But some could actually just be embedded, covalently bonded to the head in some way or shape or form. But there is no functional difference here that I know of. Okay? Good question, though. Good observation. Anything else?
Can I say what?
Yeah?
Can I say what answer for number 9?
Oh, number 9?
Yeah.
All right, what's your answer for number 9?
I put the difference between surface proteins and the membrane spanning proteins. Our surface proteins do not reach across the membrane.
Yes.
All the membrane spanning proteins do.
I think that's easy.
It took me a second.
I mean, do you agree it's easy?
I gotta check my notes for that question though.
I mean, guys, the key here is surface protein versus membrane spanning protein. Membrane spanning. Spanning means crossing. So crossing the membrane. So that it's all in the word. When a carbohydrate chain is attached to protein, what's it called? It's a glycolipid.
The protein?
Oh, glycoprotein. Sorry,
Yes.
my bad. So you have some protein, and it's covalently bonded to sugar. So I'm not going to draw it, but just so you know, you have a protein and just have a sugar on it. And the sugar can be a whole chain of sugars, right?
Yeah.
We know they can be a polysaccharide, oligosaccharide, what have you. When the carbohydrate is attached to phospholipid, what's it called? It's a glycolipid. Glyco, again, is
Sugars.
sugar. What types of molecules are shown moving across the membrane?
Small, non-polar.
Small, nonpolar.
Or small.
And we gotta go look, reference the thing. We got
Could it be both?
small,
Yeah, small and non.
well, small nonpolar and small polar molecules. You're right, they're both, right?
Yeah.
If we look at this, it is both. It's not, it could be. You look at the diagram, the answer is both. Small nonpolar and small polar. So what's the example of a small polar molecule that crosses a membrane, although it's not as easy as a nonpolar? Water, right? So water does cross the membrane, but it does it slowly. Where exactly in the membrane do these molecules pass through? In between the phospholipids in the bilayer. So when you look at these small ones are moving right in between, you see?
Yeah, this shows the example.
Right in between, they're moving out right in between them. Here, this is in between. Does that make sense? They're not using any help. There's no help necessary for these. They're moving across. How does the concentration of small molecules outside compare to concentration inside? There's less inside than outside.
What would you say again for the answer? Less inside than outside. Okay, never mind.
So that's the answer. That anything like this would be right. So less outside would be high, and inside would be low.
Okay,
Outside's high, inside's low. That's it. Because particles move randomly, molecules tend to move across the membrane in both directions. Does the model indicate the molecules are moving in equal amounts in both directions? Justify your answer in the sentences. Does this show that molecules are moving inside and outside in equal? No, it shows more going inside than outside.
I said because.
No, it does not show this. Equal movement. It shows
More going inside than outside.
3 going in
Yeah, 3 going in.
and 1 going out. This makes sense if the movement is random. There are more outside. So randomly you would expect more to come in than leave. How many people are in this room, do you think? You keep on that phone, you're going to lose it. I'm not blind. You all act like I'm not paying attention. I do. I let you get away with little things. Maybe that's my mistake. Maybe I should just pound you. Every time you take it, I take it away. Just over and over and over again. The same children do the same behavior every day. Put the phones away and don't pull them out in class.
What'd you ask?
How many people do you see in the room? It says 20. Let's say it's 20. What if you start running around the room, just moving in every direction, bouncing off the walls, like you're a bunch of... Oh my God! Assuming you're not going to hit yourself, just you touch a wall, you move around, move the other way, you're just randomly moving, and the door's open. Would you think that randomly one of you would go out the door?
Yes.
Eventually, it might take a while, but you would, right? If there's 1,000 kids in the hallway, and there's only 20 of us in here, and that we're all randomly moving, how many do you think there'd be more often you would find some kids coming in? You'd see more kids coming in than would be going out, right? Just if you just keep randomly moving till you bump into each other, since there's more outside in the same amount of space than inside, you would expect more to come in than go out, just randomly. And that would keep happening, so you keep increasing the number in here until both sides are about equal, and then you would just get one going in, one going out, one going in, one going out, and it would just stay the same on both sides, the same concentration on both sides, but it would still be moving. So we call that dynamic equilibrium, right?
Yeah.
So, all right. When there's a difference in concentration of a particular particle on either side of a membrane, a concentration gradient exists. Particles move along the concentration gradient from high to low concentration until a state of equilibrium is reached. At that point, there is no more movement in one direction. Although particles continue to randomly move across the membrane, they often called dynamic equilibrium. The net movement of particles along the concentration gradient is called diffusion. All right. So when we talk about concentration, remember, we're talking about... Well, yes, but what is concentration? It's the number of solute divided by, I shouldn't even put number. I should put amount divided by the amount of solvent.
Isn't it a passive transfer if it goes down the gradient line? Huh?
Yes, we'll talk about that in a minute. Okay. But whenever you have any movement from high to low, we call it diffusion.
Yeah. So
it's called diffusion, all right? And it just means net movement.
Which is the minus.
All right. Look back at model 1. Which particles are moving by diffusion across the membranes? The small polar / nonpolar,
Yeah, both dropped off.
right, molecule. So the key here is the smaller the molecule, the more nonpolar it is. The smaller the molecule, the more nonpolar it is, the easier it is for it to pass the membrane. No one should be having their computers out in this class right now. Everybody should be writing notes with the hope that you would then study these notes, revise them, recite them, and be ready for your exams. Yes. Excellent. There is no difference. Except that osmosis, so the reason I say there's no difference is that's what diffusion is, net movement from high to low, right? But osmosis, no. Osmosis is diffusion of water. So water's high to low. All right, going from water, where water's the highest to where water's the lowest. All right. Is that good? Good question, by the way. All right, so let's move on. It's fine. Number 5, page 5 here out of 12. Using all the information in previous models and questions, circle the correct response to correctly fill in each blank. Diffusion set is the net movement of molecules from an area.
From high.
High is correct.
Low concentration.
To an area of low concentration. There you go. The molecules will continue to move along this
Concentration gradient.
concentrate.
Until they reach equilibrium.
That's right. Concentration gradient until they reach equilibrium, and we call that dynamic equilibrium.
Was kind of confusing, but
Once equilibrium is reached, molecules will continue to move across the membrane randomly, and not in one direction though. In both directions, they would randomly move in, and they're always moving randomly. It's just sometimes there's so many more on one side that more move in one direction than the other. Facilitate diffusion. Now, facilitate what?
diffusion.
Diffusion. What is diffusion?
The movement of concentration of high to low.
Movement from high to low with no extra energy.
Yeah, no energy is needed.
It's a tricky question because some energy is needed. That's why you have kinetic energy, but no extra energy. Extra. Facilitated just means help. With help. So any of these channel proteins are examples of facilitated diffusion. Here you have a hormone like that. If the hormone's bound to it, then the channel opens. You bind, the protein binds to it on the outside. This part's obviously outside, right? Glucose is here. If the hormone binds to it, it opens the channel. And how are things going to move? They're going to go from high out here to low. So when you eat sugar, you have blood glucose out here. This is the blood. These are the cells of your body. Your hormone binds to it. It opens the channel. And the glucose flows through easily. Yeah.
When the hormone goes to the hormone binding site, doesn't it kind of work like a key to open the channel?
Yes, it does. It works like a key. And what happens is that the hormone binds, but it doesn't covalently bond. So it's not a chemical reaction. They associate very much like a salt, sodium and chloride. They come close to each other. That pull, intermolecular forces pull, it changes the protein, literally pulls the protein apart. So you have to think of these proteins not as like a wooden door that can't be forced, but think of it like a bouncy house, air semi-filled space that you can just kind of squeeze on through, push it open. So it pulls this binding to this, changes the shape of the protein. That changing the shape opens a space that allows stuff in. So this is not, when we use the word doors and windows, we think of these hard things that we open or close, but they're more about changing their shapes. The shapes change, and where you used to have something that would block and it would be closed, now is open. Yeah.
So you said they're always moving randomly. So
How
like
does
when
it?
the channel opens, because you see in the model, it's kind of like it's moving in a straight line.
Yeah. It would. You're right. It would go both directions, but since there's so much more out here, this is called the net movement. So there's
So would
mostly
they be
moving it.
in a, like would they actually be moving in a straight line like that, and then
They would just kind of not in a straight. They would just kind of like wiggle around and find their. It's a really good question. I like the way your brain works. You're like trying to figure this out. You're trying to see it in your mind, visualize it. They're just showing you the direction of the movement. Now, I got to say, what the problem is, your cells don't want to let it back out.
Yeah, it just.
So hold on, think about this. If you left it open, what would happen? Mostly it would come in, and then as soon as it could, it would go back out again, just randomly, back and forth. What your cells do as soon as sugar comes in, it adds a phosphate to it, boom, traps it. We're going to get back to that when we go over glycolysis. But as soon as it comes in, your cells trap the sugar. So it can't get back out again. Yeah.
So, you know how she said, like, what is moving randomly, right?
Mm-hmm.
For the hormone binding site, like, does it, like, how does it get to it? Is it, like a magnet?
They're like magnets, yeah.
To the thing?
Yeah, they're kind of like magnetically stuck. They don't really covalently,
Like,
they don't.
When the glucose is trying to get in, like, and it all moving, like, the glucose, the hormone, like, how does it, like, know to go there, like, it goes there? After it is moving randomly in all directions.
How does the sugar know?
Yeah, the hormone, how does it, like...
How does the hormone find the site?
Yeah.
Excellent question, randomly. So the more hormone you release, the more these molecules can find the site. But they have to be specific. See how they're specific?
Yeah.
They have to fit. They have to. It's you said it earlier. It's like a. It's actually much more complicated than this. The shape is much more complicated. It's much more like a key. You're right. The drawing is like two curves, right?
Yeah.
But this is actually a very complicated. If you look at your notes from the membrane notes that I gave you, you can see how complicated these proteins look. Their shape are very, very important.
That's why
Right.
I put it, like, kind of works like a key down
It is. It works like a key in the lock. Yeah.
And then I was gonna say like, so I remember in the beginning of the year we were talking about homeostasis and stuff.
Yeah.
So as they're moving through the channel and then you said they get trapped. Does it know like when to stop? Like,
No, it'll keep trapping them. That's why when you eat sugar, when you eat candy, at first, you need to get the sugar out of your blood, so your body releases insulin. It binds, it opens them up, and it lets all as much as it can into the cells, and it traps all the sugar. It doesn't let anything back out. So it wants to keep homeostasis, right? So after a while, what happens? Your blood sugar level gets real low because you're trapping all the sugar inside your cells. So you send another signal out. You stop the insulin. You send another signal out called glucagon, and that tells the cells, stop this. So it gets these channels out of the membrane, and it says release sugar from your liver, and it releases the glycogen from your liver in the form of sugar to rebalance that sugar level. So you're constantly doing negative feedback back and forth, keeping the sugar level at a net stable.
So then once that signal is sent, the channel is gonna close, right?
Yeah, once the glucagon is sent, this channel does not only closes, they take the channels right out of the membrane. So this is a little simplified because insulin actually signals, it's a little more complicated, it signals your cells to release the channels. They're stuck in vesicles. And when you release that signal, releases the channels go into the membrane and they allow sugar in, sugar gets trapped. Then when you get too low, they take the membranes out, so that's why we call it a fluid mosaic model, because your membrane is constantly changing. Yeah.
So what if, like, the channel was left open, kind of, right? Like
Then you would die because you have low blood sugar, you pass out, go into coma, and die.
The glucose keeps on going in, and like, what is like too much on like the inside, like the inside?
Yeah, it's never, once you get, hey ladies, focusing, it may be time to leave, but they're asking really great questions over here, you should listen. If you have too much sugar inside, your body makes fat with it. Your cells make fat.
Okay.
Yeah,
So is that why?
I can't hear her. Is the example what? You guys are being so rude right now. Could you wait till her question's done? Please say again. Yes, it's an example of stimuli response. Correct.
So
Correct.
you said after a while, the sugar starts mixing with fat. So like
Not mixing it. It gets turned into fat.
So like is that like when you intake too much sugar? Like,
Yeah, when you take too much sugar in, what do you do with the extra calories? You turn them into fat.
One more question.
Huh?
One more question.
Yeah.
So, anyways, so if it left open, right, and then it's like going really like after it's like too much, really come out like some dynamic equilibrium on both sides.
It would, but your cell, what does your cell do as soon as the sugar comes in? It doesn't close, it traps it.
Traps it, or the
So the sugar only fits through because it's like a triangle. But what your cells do is it adds a phosphate on it and it changes the shape so it can't get
And
through
then it
again.
traps with the phosphate.
It traps the sugar inside, and then it processes it. It turns in all that sugar into ATP.
Yeah.
And all the mass of the sugar, any candy, any food you eat, the carbon in it, you breathe it out.
It's an ATP and then I triphosphate.
Triphosphates, correct.
Can you give my paper?
Yes. All right, we'll finish this on Monday. Yeah. So they labeled it membrane gated spanning protein or channel protein, and you see here's the gated. That's actually labeled here, gated channel. What type of molecules attach? What type of molecules attach the protein, or do attach to the protein? We call them hormones or signals. Again, I don't see any difficulty here except reading hormones or signals. We covered signals earlier in the year. Explain in detail what happened to allow the glucose molecule to pass through. The signal, the hormone, bound to the protein and changed the protein's shape, allowing sugar to come in. It says, read this. So some molecules such as glucose use gated channels as shown in Model 3. However, not all channels are gated. Some channels remain permanently open and are used to transport ions and water across the cell. To facilitate means to help. Explain why this type of diffusion is called facilitated diffusion. Proteins help molecules and ions cross the membrane. The tails of phospholipids are nonpolar, therefore they do not readily interact with charged particles such as ions. How can this explain why facilitated diffusion is necessary for the transfer of ions such as sodium and potassium across the membrane? It creates a passageway that is hydrophilic, which again goes back to the membrane that a lot of you did not put too much effort in. If you did not understand, if you didn't color it correctly, you didn't think about what those channels you were doing. The reason I had you color the center of the membrane one color and the outside of the membrane a different color was because I wanted you to try to understand that this is what allows ionic substances through, the fact that you have this space, this part of the protein that is hydrophilic that will allow protection from the hydrophobic repulsion of the lipids. So active transport, that's something a little different. It's the word here is active. And so now things get a little more complicated if you didn't really understand passive transport, where things are moving from high to low. In active transport, you're moving from low to high. It's the opposite. So if you understood passive transport, that's easy. Here you can see the example. Of course, if something's active, anyone's active, it needs energy. You need energy if you're active. And lastly, the energy is found as ATP. the energy's form is ATP. So those are 3 things about active transport you need to know to be able to identify it as active transport. So here you have a protein. There's some outside, some substance that needs to be transported outside, a lot more inside, but you need to get it out of the cell. You want to move it against the concentration gradient. If you want, not out of the cell, you want it in the cell, excuse me. If you just opened a hole, if you just open a gated channel, then this substance would just go right out. That's diffusion. That's passive transport. But you were trying to get it to go from outside in. You're trying to create a higher concentration inside than outside. Higher than what it is already. You're trying to get these 2 in, not to allow these out. So to do that, you have to use ATP. ATP has to bind. It allows for a conformational change, a change in the shape again. But this time, instead of just making a hole, it allows the protein to bring the ions or the substances in. So it comes in, and things go from low to high. But in order to do that, you need ATP. You need the energy released from ATP turning into ADP, which we've gone over already and you were quizzed on. So which part of the cell membrane is shown in more detail in Model 4? Look back at Model 2 if you need to. Again, the membrane spanning proteins. Again, it's a membrane spanning protein that is a gated channel. Or it's gated, voltage gated. What type? And actually, it's not even gated. We have to what it should label, and let me go ahead and correct that now. It should be labeled a pump. I'm not sure. You should all know what a pump is. When you have a bicycle tire pump, you're taking air outside and you're pushing it from low to high. Obviously, the tire pressure is higher than it is outside. You pump it, you push it into the low, use energy to move it from low to high. So that's what pumps do. They move things from low to high. So it is a pump. What shape represents the substance being transported across a membrane? I hope that's easily a diamond to you. I don't know what other shape you'd call it. In which direction is the transported substance moving? You're going from low to high. It says from an area of concentration to an area of low concentration, or from an area low to high. It's low to high. There is more inside than out, and the material is going in. And ATP is used.
Does the D stand for double?
Where do you see D?
ADP?
Ah, okay.
Triple?
Yeah, it's a, yes, in a way, yeah. It's di, but yes.
Okay.
So adenosine is, ATP is adenosine, the nucleotide adenosine, with 3 phosphates attached to it covalently. So it's adenosine triphosphate. And ADP is what's left over. When you still have adenosine, that doesn't change. But you only have 2 phosphates on it. And it's adenosine di, that's diphosphate. So tri and di, double and triple. All right, moving on. Again, we're on page, what is this? What page number are we on? 6. Next, is the substance being moved along down the concentration gradient? No, it's not. No, it's going against. I don't know why anyone would have a trouble with this. Is it going with the high to low, or is it going low to high? If it's going high to low, that is going with the concentration gradient. This is going low to high, so it's going against the concentration. It's going from low to high. High to low would be going with the concentration gradient. ATP is a type of molecule that can provide energy for biological systems. Explain how the energy is being used in Model 4. Phosphate, a phosphate bond is being broken releasing energy. What type of, what happens to ATP as it binds to the protein? It loses a phosphate and energy. So it becomes less energetic. What type of transport is shown in, the type of transport shown in Model 4 is called active transport, while diffusion is facilitated, while diffusion and facilitated diffusion are called passive transport. Given the direction of the concentration gradient in active and passive transport examples, explain why active transport requires energy input from the cell. Because material will not randomly move from low to high. Energy must be put in. So there's many examples we can use. We've used a number of examples in the past, but if we have, I just used a bicycle tire example, so might as well go ahead and look at a bicycle tire. If you're trying to pump air into something that's high pressure, which means all high pressure means is there's a lot in here in a small space, and you want to put more in there, you better be ready to push. You need to pump. You better be ready to push. You better be ready to pump. So you need to push energy in. So it requires energy input by the cell. Active transport does. So yes. Yes. No. No. Molecules move along down the concentration gradient. No. Yes. Yes. Moves molecules against or up the concentration gradient. Yes. No. No. Always involves channel membrane proteins. Membrane spanning proteins. Yes. No. Yes. Molecules pass between the phospholipids. No. Yes. No. Moves ions like sodium and potassium. Yes. No. Yes. Moves large molecules. Yes. No. Yes. Moves small nonpolar molecules. Yes. Well, not active. No. Yes. No. All right, that took you through page. So that being said, that this is no for active transport, yes and no. I want to qualify these answers because the answer really should be maybe and maybe. They are used for an inactive transport to move. For instance, aquaporins is an example. Aquaporins. Aquaporins move water, a lot of water, from high to low. They're passive transport, so they're a form of facilitated diffusion, and they are small polar molecules. They can pass through the membrane without help, but cells do use aquaporins to move them much more quickly through the membrane without help. And even though they're small and non-polar, I'm not sure if I can't think of any examples of small non-polar molecules or polar molecules. Well, actually, no, I can. So this is definitely a yes here as well. So the best answer would be yes, not no, but yes and yes. Though I can understand why people would say no. Because aquaporins would be an example of a facilitated diffusion moving water, which is small and polar. And another example of an active transport would be in prokaryotes and protists, you have something called a contractile vacuole. That what it does is it fills up with water. And when then it uses ATP to contract and shoot the water out of the cell. And the reason it does that is because unlike humans and animals, plants or rather protists, again we call these protists, have to swim, have to move. They exist in varying concentrations of water, and because of that, the inside of their cells can fill with too much water, and they need a way to get rid of it. So they have this contractile vacuole. They have a nucleus. They have a nucleolus. They have a endoplasmic reticulum, a rough and a smooth. They have ribosomes. They have mitochondria. They actually have a cell wall, as we've discussed before. These proteins. They have a cell membrane. Then they have a cell wall along the outside of the cell membrane to help protect them. But they need a way to get rid of extra water that might come in for various reasons in concentration of H2O. They need a way to shoot that water out. And I'll show you a video in class of them doing that. So that is active. It does take ATP. It is going from low to high. And it is, it does use ATP. So that is a form of active transport. It does use, and again, aquaporins, which can be found in various creatures, are an example of a facilitated diffusion with water. Moves large molecules. Facilitated diffusion, large is relative as well. Really large things, it is an active, either way, it's active transport. Facilitated diffusion, moving large molecules. Anytime you're looking at, either ionically charged or large molecules, you're going to need some kind of facilitated diffusion. They're not going to move through the membrane easily. That's true, but what gives me pause here is the word large. Really large molecules have to use a form of active transport. But there are larger molecules that can use facilitated diffusion. Of course, diffusion, simple diffusion, another word for diffusion, simple diffusion, requires the molecule to be small, polar or nonpolar, because it has to move in between the membranes. And all the rest of these are pretty self-evident considering all the things we've talked about thus far. I'd expect you to have at least that much. Okay, so looking at this graph, some people had trouble with it, and it's analyzing a graph. So in general, people can run into issues. Let's go ahead and start looking at. There is no title to the graph, so let's go ahead and look at the x and y axis. It says concentration difference. So as you come up, the difference, and so again, I should start with what I like to do is draw things out. So I draw a cell, and this is in, and this is out. And so, and I could have a concentration difference that's large between in and out, or that's small. So maybe this is 90% inside and 10% outside. And this is 40% concentration inside and 35% concentration outside. So there's a difference between they're both high inside and low outside, but the difference here is a good 80%, and the difference here is only 5%. So you can see that this one would have a higher difference. That's what they mean by concentration difference. The bigger the difference, you can see with diffusion and facilitated diffusion, it's the difference between the two sides that tells you how fast the material can move in and out of the cell. Why is that important? Because you can't wait too long to get some of these functions to occur. Imagine you had to wait 10 minutes to get oxygen into your cells. Your cells would die. So you need to have to be able to do it quickly. So keeping a concentration difference, making the difference big, helps you move it quickly where you need to go. So in any case, you see with diffusion and facilitated diffusion, the more the concentration difference on both sides, the faster the concentration, the movement. Diffusion, there is no limitation. Facilitated diffusion, there is. And we're going to look at why in a minute. You'll notice that it keeps going faster and faster until it reaches a certain point, and then it plateaus, and that's all that can happen. Okay, we'll see why that's important in a minute. With active transport, the rate is constant, and there's a good reason for that. Where diffusion, the bigger the difference, the faster it goes, and there's doesn't seem to be a plateau on that. Let's see why that's so. So, and this was the challenging part I had told you about. The first few pages should not have been that challenging. So if we draw out a membrane with different conditions, let's take a look at the first one. This is what we would call simple diffusion. And what you see here is that this is high, not much, in fact, zero over here. You can think of it as maybe a cell coming into the lung, in the blood vessel, coming to the lung to pick up oxygen. Almost no oxygen inside the cell. A lot of oxygen relative to inside the cell. Oxygen diffuses, being nonpolar and small, diffuses through the membrane, goes right into the cell. The higher the difference, the faster it happens. These molecules can come in anywhere. So the only thing that they have to worry about is how much surface area and how much membrane there's space for these molecules to come through. And then the other thing it has to worry about is what's the difference between the two sides, high and low. If it's a small difference, it's going to be slower. If it's a big difference, it'll be faster, right? That should make sense, since there's only two things impacting this. The surface area, how much space is there for it to cross the membrane, how much membrane there is, and two, the difference in concentration. All right, so simple diffusion. So when we look at this simple diffusion here, or diffusion, again, this really, you're going to see it called simple diffusion, so you might as well get used to that. Simple diffusion, just assume the higher the concentration, given a certain cell, given a specific surface area, whatever that means, like so. If there's two cells, if all the surface areas are the same, then only the concentration difference is going to be an issue. All right, so simple diffusion, that makes sense why that's a straight line. Why is facilitated diffusion, why does it keep going faster the higher it goes, right? Because surface area is still an impact in facilitated diffusion, because the more membrane you have, the more space it has to get through. But then on top of that, the higher the concentration, the faster it goes. That also makes sense because, again, you have more stuff outside. They're bouncing around. More stuff gets across the membrane easily. But why does it plateau? Why does it stop going up the way simple diffusion does? Well, the answer is that with facilitated diffusion, it's not just the surface area of the cell, because you don't have the whole cell, right? You don't have the whole membrane like you do in simple diffusion. In simple diffusion, the molecules move in between the molecules of phospholipids. So the more phospholipid, the more cell surface you have, the more quickly things diffuse through. But in facilitated diffusion, which is what we call this, facilitated diffusion, you're still going from high to low. So it makes sense that you would expect the higher concentration to come through quicker. But this time you don't have the whole membrane. You only have these facilitators, right? You only have these membrane spanning or transmembrane channel proteins. That's all you have. So they can only come in these spots. So that'll go faster the more you have outside, the bigger the difference in concentration between outside and inside, the faster it's going to go through until all the channels are filled and they're moving as fast as they can through these channels. At that point, you'd expect that it's not going to go any faster. So it keeps going until it reaches the limit. That limit, this is the limit, you see. When you hear this word limit, that's as fast as it can travel. That's the maximum speed that you're going to be able to get stuff in here, because it can only come through these channels. It can't go through the entire membrane the way it can with simple diffusion. So in simple diffusion, it keeps going faster the more constant the concentration difference, where the facilitated diffusion, it can only go as fast as the channels allow. Now, why is active transport, first of all, high, and then it stays high? It's not impacted at all by the concentration difference, because concentration doesn't matter for active transport. First of all, active transport is going to move from low to high. But the difference doesn't really matter to active transport. The only thing that matters to active transport is that you have these proteins, you have ATP. As long as you have ATP, these will keep grabbing and moving. So really, it's not impacted at all by the difference in concentration, whether it's low here, high here, low here. What it is impacted by is how much ATP you have and how many of these transporters you have, which is not on that graph. So when we look at what are the steps of active transport, you have this transporter here. It binds the molecule. When it binds, the second thing happens, ATP is used. It leaves behind a phosphate. That changes the shape of the protein. Notice the change in shape. The molecule gets taken into the protein and it continues to change in shape, and then it kind of spits out the molecule on the inside. And then the ATP is the phosphate that was taken off ATP also leaves and is here in the cell, and then the protein goes back to shape, normal shape, waiting for it to another molecule to take in. So that just keeps going round and round and round and round, and as fast as it can. As long as you have ATP, it'll keep bringing these molecules in, and so concentration doesn't really impact active transport. So this actually makes a lot of sense. Active transport is going to move quickly. It's just going to use energy, so it's a little harder. It's so by definition it's going to be harder to do. Active transport is not really impacted by the concentration difference, since the only thing that impacts active transport is how much ATP, how many transporters you have, and how much ATP is you have for them to use to move the stuff in and out of the cell. So this graph makes really beautiful sense when you think about it. Which type of transport would be best if the cell needs to respond to sudden concentration gradient difference? Explain. If you had to respond quickly to concentration difference, notice they didn't say a large concentration gradient, they say sudden. It would be active transport. So active transport is going to be the best because it's fast all the way across. It's not going to depend on the concentration difference. It's always a quick transporter. So if you have a sudden change in concentration, you want active transport to quickly move things in and out.
You can't tell with your haircut, you have like 5 weeks.
That being said, simple diffusion, if there's a large difference, simple diffusion would be nice. But if there's a small difference, it would be hard. A small difference in this between the concentration differences on each side of the membrane, then passive transport, any kind of passive transport would be slow. But active transport's always high. So given these conditions, I would say active transport, because it is a fast transporting method, always, where the others depend on concentration. Why would the line representing facilitated diffusion leave off the concentration gets higher while the line representing simple diffusion continues to go up steady? We just explained that, and that's because there's a limit to the number of transporters facilitators in facilitated diffusion. Where diffusion is just simply the surface area of the cell membrane in total, and the concentration difference. So again, those are the three big ones. So here we have a simple drawing as the sugar molecules on the outside. You see here, it's large. Water molecule is small, it's a solvent, and sugar molecule is a solute. Selectively permeable membrane is here, all right? You see water going in, you see water going out. No sugars going in and out. That's what I see. Solution consists of a solute and a solvent mixed together. For a solution, model 1, identify, provide this symbol for the solute. The solute, I don't know why anyone would get this wrong. Solute is labeled, it's a circle, and the solvent is water, it has a plus. Consider the size of the sugar molecule and water, model 1. Which molecule in diagram model 1 would move through selectively permeable membrane? One, we have to say, I think it's clear it's going to be water because it's small, where sugar is big. Consider the arrows indicating movement across the membrane. Which direction are water molecules moving into and out of the cell? Well, it's moving in both directions, right? And if we look at it, let's count them. We got 1 going out, 2 going out, 3 going out, 4 going out. So 4 going out and only 1, 2 going in. Okay. I'm going to go ahead and cut to the chase and draw this. Now, let's do a concentration calculation quickly. Here inside you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. About 23 of the solvent on the inside. (23) solvent. And how many solutes on the inside? 1, 2, 3, 4, 5, 6. 6 total. 6 solute. Outside, we can count. We see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 solvent. And solute on the outside. 1. Did I count that right? Yeah, solute. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 solute. So outside the concentration is 1, and inside the concentration is, so outside the inside the concentration is 0.2608. Outside it's 1, right? Solute and solvent are equal. Okay. So obviously outside the solute concentration is higher than inside. I think that's fair to say. So consider so if it's higher outside, so I'm going to go ahead and mark this as high and inside as being low, we'd expect things moving from high to low, if they could move. So this is high to low solute concentration. But remember what we said. We have to divide this inside space into 2 parts. 23 of which are water, or 26%, which is a little more than 25%, something like this. This is mainly water on the inside. If we look at the outside, we have exactly 50-50. 50% water and 50% solute. So even though the solute concentration is higher outside, the water concentration is 50% of the outside, right? Where water inside is 77%, something like that, on the inside. So water is actually a higher concentration. You can see it here in blue. There's more water inside than outside. So therefore, for water, inside is high, and water outside is low. Now, that being said, the solute cannot pass, but the solvent, in this case water, can. So water is going to diffuse, but how does diffusion happen? The diffusion of water, remember, is osmosis. Osmosis, diffusion of water. Water happens from high to low. Diffusion happens from high to low. So you'd expect most of the water, the net movement of water, to be from inside to outside in this case. So we expect most of water going out. And in fact, we see 1, 2, 3, 4 going out and only 2 coming in. So that's what we see. The net movement's out. That's because there's more water relative to solute inside than outside. All right, so that's the theory behind it. Let's go ahead and keep looking, keep applying it. Consider the arrows indicating movement across the membrane. In which direction are water molecules moving into and out of the cell? All right, so both in and out, which we expect because it's random movement, diffusion is random movement, are more water molecules moving into or out of the cell. Out is the net direction water movement in or out of the cell. Out, which again we expect because of everything I just explained. Circle the correct word to indicate the changing concentration of the sugar on each side of the membrane as water molecules move. So as water keeps moving out, this goes from a concentration of 1 to smaller and smaller number, right? It starts with 1 because there's 15 solute, 15 solvent. But once we add 3 more, that would be 15 solute. That doesn't change because it can't pass the membrane. But now solute becomes 20. So it's 15 divided by 20. So if instead of 15, this became 20, then it would go from 1 to 75%. So 1 to 0.75. So this is going to get smaller and smaller outside. The concentration of solute over solvent is going to get smaller and smaller. As more solvent comes out, you're dividing by a bigger and bigger number. So what happens is the solution inside the cell will become, the solution inside the cell will become more or less concentrated. Well, as you leave water, as water leaves, the number of solutes don't leave, but you're losing more and more water. That means this number is getting smaller. This one stays the same, so this number gets bigger. So water, the concentration inside gets higher. And the solution outside will become, instead of higher, because that might be confusing. So let's just use the word more concentrated. So more, and outside can become less concentrated. Applying what you already know about the random movement of molecules, what will eventually happen to the concentration on both sides? They will become equal. The definite diffusion is the movement of water molecules across the membrane from an area of high concentration to low concentration. According to this definition, is the model cell in Model 1 undergoing diffusion. So what we have is this diffusion happening? Well, the answer is yes and no, because it's not diffusion, because it's not the solute that's moving, it's the solvent. And let's be clear, water is diffusing, moving from its high to its low. So it is called osmosis. So osmosis is the diffusion of waters, and that's what's happening here. In the cell diagram model 1, where is the higher concentration of water, inside or outside the cell? And I believe we said the higher concentrations inside, and you saw me draw it out and count them for you. Is a cell in model 1 undergoing diffusion if you consider the concentration of water on either side is selectively permeable membrane? Is it undergoing diffusion? In a sense, yes, but I'm going to just go ahead and say no, because we do make a distinction between diffusion and osmosis. So in a way it is, because osmosis is the diffusion of water, but in essence, no, because diffusion is when the solutes move. Osmosis is the movement of water from high water concentration to low water concentration across the semi-permeable membrane. And there's some models here, and we'll finish as the class is coming in. We'll have to finish the next pages in a minute. So here, before I go on, let's take a look at what's happening here. You have the words, as I mentioned on Tuesday or on Monday. There's three words here, and when you're talking about the root word, or part of the word here being tonic is talking about the stuff that is the solute concentration. Hyper means high, and tonic is solute concentration, or solute. So high solute, hypertonic. Isotonic, iso means equal, and tonic again is solute. Concentration. And hypo, tonic, hypo means low, and tonic means solute concentration. So those are the three conditions. When you put all these high, equal, and low are relative inside to outside. So what that means is that inside versus outside is hypertonic. When something's isotonic, the inside solute concentration is equal to the outside. And hypotonic means inside you have less, or rather outside you have less solute concentration than inside. Now that being said, I keep pushing this because you really have to think about this. If it's high solute concentration outside, let me zoom in here. High solute means low water. And of course, iso means equal. So they're both equal there. And hypo means outside, low solute, which means high water. Now, if water is high outside and the solute cannot go through the membrane, then water's going to rush in. It's going to keep going in until the cell bursts. We call that lysed. Lysed. And we talked about lysing, like lysosomes. So this should not be new to you. And so these cells are lysed. They're broken. They're ripped open. The balloon popped. Normal's normal. And when they shrink, we call it creatined. Shrinking up. Obviously, they're about to die, so it's not good. Now, interestingly, with plants, they have that thing called a cell wall. That's very useful. You'll notice that this is the cell wall, this is a cell membrane, this is a cell wall, that's a cell membrane, and this is a cell wall, that's a cell membrane. Here's this central vacuole. As you get more and more water, the water leaves the cytoplasm and goes into the central vacuole So the central vacuole acts like a big storage area of water for the plants. So when we look at hypertonic conditions for a plant, you're talking about high solute which means low water. Here again, they're equal. And here we're talking about low solute, which means high water. So here you'd expect water to rush in, and it does. But luckily, plants have these cell walls that help keep. These central vacuoles are going to get filled, which means everything's going to get pushed out to the side. Poor animal cells like you and I, our cells don't have a cell wall, so when they get overfilled, they burst. These plants get overfilled, but they don't burst because they have a cell wall that keeps them all together. We call that turgid. This is a normal, and this is plasmolized. Plasmolized is when the cell shrinks up and dies. So plasmolized is the same as crenated, and of course, plant cells don't lyse the way animal cells do. So those are kind of the three conditions. If you don't want to try to figure out where salt is, where water's higher concentration, lower concentration, then just consider if they're in a hypertonic solution, which means outside higher solute, in a hypertonic solution, this is the solution out here, you put the cells in it, then you'd expect water would rush out because high solute, low water, high to low water, that's the way osmosis would flow. Assuming the solute cannot move. All right, let's move on. Now I'll answer these 2 pages and then we're done.